Starting. Yep.
Fabulous. Okay, welcome everybody that's joining us from various locations today. We're going to start this webinar so that we can keep it moving. It's a pretty jam-packed full session. So I just want to keep it moving if we can. My name's Karen Thomas and I'm with Melbourne Water, and we're delivering this project as part of our carbon farming outreach program. Just going to share a couple of quick slides just to do a bit of an introduction to our guest speakers today. Tina, just let me know if we can see the screen, because I know we've got a few lag things happening today. So you can see the screen. Great. I will just put it into presenter mode. Just let me know when it's in presentation mode. There we go. I think that's right. Okay, so welcome everybody. Just like to acknowledge that I'm on the traditional lands of the Bunurong people here in the Mornington Peninsula, and I pay respects to elders past, present, and emerging, and welcome any Indigenous people that may be joining us today. Just want to let everybody know that we've disabled the chat for this session because there's quite a few people planning to join, but there is the Q&A. So if you've got any questions, please put them into the Q&A and we will go through them as best as we can at the end with the time that we have left. Just a heads up that we may run a little bit over, but feel free to log out if you need to. The session is being recorded and you can watch it back afterwards. So we may have a little bit of extra time for the questions at the end, just depending on how many questions there are. But I just wanted to allude to that up front. Just going to introduce the speakers now. So we have Joel Williams joining us today from Canada. And Joel is an independent plant and soil health educator and runs a business called Integrated Soils. Joel's a globally recognised authority on soil biology, plant nutrition and agroecology. Joel's based in Canada and has done extensive work across Australia, Europe and beyond. So welcome today, Joel. Thanks for joining us from Canada. Here it's 0, almost that cold in Melbourne today. Our second speaker, just a heads up, we've got Dr Mary Retallack. Unfortunately, Mary is very unwell at the moment. And so Mary has done a pre-recording for us that she has managed to do. So we are going to play her presentation as a recording. So please don't drop off because she's not here. She's here in spirit and she still has a great presentation. And any questions that are for Mary at the end, we will direct to her and she will endeavour to respond to them in the new year. And we also wish Mary well as she goes through her convalescence period at the moment. Mary's a third-generation viticulturalist and a PhD qualified agroecologist. Mary leads the National Eco Vineyards program, which has been partnering with Wine Australia to promote biodiversity, soil remediation and ecological pest management across over 140 growers across Australia and a number of wine growing districts, of which we had 2 here in Victoria previously with the Mornington Peninsula and Yarra Valley. So I'm going to welcome these speakers. We will listen to Mary first, and then we will have Joel as the second speaker. And then Joel, because he's available and here live, Joel will also be able to tackle any of the questions in the Q&A that are relevant to his presentation. So I'm going to stop sharing and just get Tanvir to set up the pre-recording. Let us know if you're having any audio issues, but hopefully it plays well for you.
And see Mary's pre-recorded webinar. Just bear with me while I do that. You can see the screen. Yep. All right, I'm going to play it now and I'll need a thumbs up again to let me know that you can hear the audio.
I'd also like to acknowledge all of the funding partners that have made today's webinar possible. I run a national program called Eco Vineyards, and if you'd like to do a deeper dive and find out more about any of the topics that I'm covering today, please check out ecovineyards.com.au, and there's a wealth of knowledge and background materials there for you to explore. By way of introduction, I've worked as a viticulturalist and also an agroecologist over the last 30 years, and both within Australia and internationally. And this combination of grounding in ecology, but also viticultural practice gives me a slightly different perspective. So I'm happy to share those insights with you today. Of course, there's many current challenges, but being the optimist that I am, I always like to see them as opportunities. So we'll have a look at what those opportunities are today. We know that we need to be really mindful of any of our synthetic chemical application. It's estimated that much of the sprayed insecticides and herbicides miss their intended target species. And we need to be really mindful not only of those perceptions from our customers, but also the unintended damage that we might be doing to our agroecological systems. So I'd also like to get you to consider some of the opportunities, and rather than going to work thinking about what can I kill today, perhaps consider what can I birth or bring life to today. And from an ecological perspective, we know the functionality of the system is really important and can also create very important buffering capacity in health of our plants, as well as the overall ecosystem. So just a brief introduction to the Eco Vineyards program. It's been running over the last 6 years in a range of different formats, and we focus on 3 key areas, including soil health, ground covers, which include cover crops, and also functional biodiversity. We've also put together a range of materials very practically focused for growers called the Best Practice Management Guides. They're available for free for you to download, and we have hard copies available for purchase at cost for those that like to read a hard copy as well. The Best Practice Management Guides touch on soil health, both the chemical and physical properties, soil biology, ground covers, and also functional biodiversity, with a focus on Australian vineyards. But all of this information, of course, is applicable to a broad range of production systems. We've been working across Australia, everywhere. We started in South Australia, but across in Margaret River in WA, in New South Wales, in the Hunter Valley and Orange. In Victoria, we've been working with Karen in particular in the Yarra Valley and also the Mornington Peninsula, and then also a range of 9 other regions within South Australia. So I'd encourage you to think about how this information may apply to your region, and also cross-sectorally, whether it's broadacre, production, horticulture, or other. So the eco within Eco Vineyards stands for ecology, and of course everything's interconnected, and we're particularly interested in the functionality and the resilience of our agroecological systems. So however you choose to grow, we suggest ecological practices that can be incorporated to create incremental step change and give growers the confidence to incorporate these practices. And we find this is the quickest way to accelerate meaningful practice change. So our measure of success is, do your practices bring you closer to or further away from the goal of growing in harmony with nature? I mentioned those 3 topic areas. There's a lot of information that we've gleaned working specifically with wine grape growers over the last 3 years. Everything from how we can nourish the soil and the soil microbes, and also read weedy species as indicators of soil health. So there's lots there to explore, along with covers, which includes not only, commercial annual cover crops, but we broaden out that definition to ground covers, which incorporates native insectary plants. They might be moving from annuals to perennials, and also looking at the inherent benefits of having a diversity of locally adapted species. So native grasses and forbs, woody ground covers, and also shrubs as well. And the benefits of stacked enterprises, it's possible to perhaps put into place the opportunity to harvest additional income from the mid-row as a stacked enterprise, and also heal the soil at the same time, along with the other agroecological benefits that we get through integrated pest management and biocontrol of some of our insect pest species. And functional biodiversity is looking at the life both below and above the soil as well. So we focus on a range of predators, including arthropods, which is a name for insects, spiders and mites, microbats, and also a range of bird species, specifically insectivorous and raptor birds of prey. When we consider the health of an agroecosystem like this viticulture or perennial system, it's important to consider that each species has a niche in the ecosystem and contributes to its functionality. So a diversity of species is really important, and also having 100% ground cover and functional root growth 100% of the time is really important to have a more fungally dominated soil where we have relationships between mycorrhizal fungi and those plants. And mycorrhizal fungi can produce a substrate called glomelin, which is really important to produce carbon that can be cycled but also held in a stable form within the ecosystem as well. So being able to capture as much light energy as we can by having plant coverage is really important, and driving that system and feeding, the soil biota. In terms of the plants, we can incorporate plants either in the mid-row, low-growing plants in the undervine area, adjacent to strainer posts, and you can see that we've got this lovely stand of remnant vegetation near this vineyard, which gives us some great insights to the types of plants that we might be able to incorporate into an agro-ecosystem. As soon as we're able to bring those plants in, and I have a passion for locally adapted plants, which are native or pre-European before we disturbed the system. As soon as they're flowering, they're providing pollen and nectar, and we start to then attract a whole range of predatory arthropods, so insects and spiders and mites. And in turn, they're able to provide a food source for higher order predators. So this process happens very quickly once we have the right plants in place and provide a food source. So we have some birds here. They are insect and seed-eating birds. We've got a diamond firetail and also a fantail here. They pose no threat in an agroecological system like this. They're not frugivores, so they're not interested in eating fruit. And they're actually contributing to the biocontrol of many of the insect pests as well. We also have microbats, as you can imagine, micro in nature, anywhere from a few grams that could fit on the end of your finger through to a small mouse size. And they're voracious eaters of a range of insects that they can catch on the wing, so moth species, especially tortricids or the leaf rollers, but also a broad range of different insect species as well. We also have raptor birds of prey like this bobal cow. They're great to keep rodents under control. You can see this one's eating a rodent in its beak. And they may also, knock out some of the lower order predators, but it's all part of the system. And we have here a brown goshawk. And the raptor birds tend to be quite territorial, and they can help to patrol the perimeter of the vineyard and keep some of those pest bird species out. So they're all really important contributors within the functionality of the system. So we're also really interested in the resilience of the system, and this describes the system's capacity to reorganise after local disturbance, and this might be also an extreme weather event or even a biodiversity incursion. So we're helping to future-proof production. You'd be all familiar with the concept of biocontrol. Here we have a predatory shield bug, Achilles schellenbergii, consuming a leaf roller larva. And that's an example of their functional trait, which is as a predator. And the parasitoid are wasps, which will lay eggs into their host. This is Delicagenetia tasmanica, or dolly for short, which is parasitising a leaf roller larva up to the 3rd instar. So the parasitoids are really important, and it's also important that we don't knock them out by using high sulphur rates. So we try and keep sulphur rates below 400 grams per 100 litres. And these are examples of conservation biological control. If we plant species that provide habitat, then we can attract them and they can contribute to biocontrol for free, if we provide a suitable environment for them. So we also know that biocontrol is estimated to provide between 5 to 10 times more control of pests than pesticides alone. If you're interested in IPM and you'd like to find out more about biological control of a range of different grapevine insect pests, we have a range of fact sheets that you can find on the Eco Vineyards website, and we've also got one on snails as well. Just keep in mind that the use of synthetic fungicides and insecticides can be quite damaging on our on our predatory insect populations. So from the top there, chloropyrifos has just been banned. It's no longer available for use, down to the softer options like BT. And in the middle, you can see that sulphur can also be quite negatively impactful on not only parasitic wasps, but also ladybird beetles and predatory bugs as well. So there's millions of little insect workers in our production systems that can provide natural biocontrol virtually for free if we understand how to attract and look after them. And it's important to remember that when we kill off the natural enemies of a pest, we inherit their work. So how do we attract these insects? There's a range of different plants that I've looked at as a part of my PhD studies. And we don't want to just be constricted to these 3 plants. We want a diversity of plants. But the benefits of some of these plants include the fact that they're naturally adapted to agro-ecosystems, and more than 85% of Australia's species of flora and fauna are endemic, and many of the predators that we'd like to attract have co-evolved with these plants. So it gives us the confidence to know that we can plant them and that they're going to provide, the benefits that we're looking for. We also know from my work that we can attract more than 100 different morphospecies or visually distinct species of predatory arthropods, and increase the functional biodiversity by more than 3 times when we plant these plants in association with grapevines alone. And we also have a book called The Natural Predators of Vineyard Insect Pests if you'd like to find out a bit more. So when we're looking at the types of habitat to attract insects, we're looking for plants that provide snap, which is shelter, nectar, alternative prey, and also pollen. And, we've got a nice little poster here that shows some of the species that you might tend to find in association. So when we consider IPM, it's important to think of cultural and biological control measures first, and chemical control as a targeted approach, and a last resort only as required or only as needed. The other thing we can consider is really getting to the heart of the issue, which is looking at plant health and the integrity of plants and their capacity to be able to withstand or actually become resistant against pest and disease attack. So when we consider the role of nutrient integrity, what we find is that nutrient-dense plants are less susceptible to pests and diseases, which is an amazing concept that perhaps we may have forgotten, through the Green Revolution, but we're now rediscovering a body of knowledge, which is really powerful. So if you'd like to find out more, we have a fact sheet that will take you through a bit of a history of what's gone before. And I'll give you just a little brief overview at the moment. So one of the things that we can do is look at the Brix levels, or the total soluble solids within the plant, the leaf blade of different plants. And we can use a refractometer that we'd normally use during vintage to look at the ripening of fruit and the fruit bricks. We can actually squeeze the sap from the leaf blades, and we can actually check out a benchmark to create an understanding of our capacity as photosynthetic managers, and to have this represented as an objective measure of plant health by looking at the Brix measurements, or the total soluble solids. So I'll come back to this concept, but if we have a look at some of the economically damaging pest insects that we have in wine production, some of them include sap-sucking species from the order of Hemiptera, and they have piercing and sucking mouth parts. So you might be familiar with grapevine scale, or even mealybugs, and or in other crops as well. They, of course, excrete honeydew, and this becomes a substrate for the sooty mould, which can be a carcinogen, and there's a really low rejection threshold whenever we have this sooty mould, kind of issue within wine grapes in particular. But this also gives us some insights to their, some of the reasons why we tend to see some of these insect pests on grapevines. So we've already alluded to some of the IPM measures that we can adopt, and we can see here the long-tailed mealybug in the top left. In the centre here, we've got one of the predators, which is the Cryptolemus, or the mealybug destroyer ladybird beetle. And you can see the adult, and then underneath you can see the larval form, which is an amazing mimic when you consider that it looks very similar to the actual mealybug that it's there to consume and to be able to help manage. It also shows how important correct ID should be, because if you have lots of mealybug destroyer larvae, you certainly don't want to spray them out of your production system. Also, green lacewings, their larval stage, or the junk bugs, are really important, and they can both be purchased and distributed as biocontrol agents, but that can also be quite expensive. So if we can attract them for free by having the right habitat, then that might be a better option as well. And of course, spiders are really important predators as well. So here's the biocontrol kind of aspect. But if we have a look at what's actually happening inside the insect and its gut morphology, this gives us some really nice indicators as to ways that we might be able to manage some of these insect pests from a different perspective. So here we have some work that's really been championed by Tom Dykstra, who's an entomologist from Florida. So this is his work. And he talks about the gut morphology of many insects, which is essentially a tube within a tube. You can see an example of a grasshopper at the top of the screen there, where food goes in through the mouth, it gets digested in the gut, and then any waste products are excreted out the back end. However, when we look at some species from the order of Hemiptera, these insects with piercing and sucking mouth parts, and this includes aphids as well, we can see that the gut morphology is quite different. They actually have several different components to their gut. They have a foregut, they have a midgut, and also a hindgut, and they also have a filter chamber. So these types of insects from the order Hemiptera actually have a very specific feeding range. So they need to have very simple forms of nitrogen and very simple forms of sugars. They can't actually consume plants with high Brix. It actually gets shunted out the back end as this honeydew. So if we can get the plants objectively healthy, then these insect species can then no longer feed on these plants successfully. In fact, they can become overwhelmed very quickly and die, within several minutes. And we see candied or mummified, aphids on plants where this has occurred. There's also another really interesting body of work that's been championed by Olivier Hasson, who is a French researcher, and he's done a lot of work into something called redox. And re and dox stands for reduction and oxidation, and this comes back to really the health of the soil, its electrical charge, and also its oxidative state. He's also got a very accurate way of being able to measure when the plant is susceptible, and the soils, the relationship with the soils has, I guess, a profile, a nutritional profile, but also a redox and pH profile, which makes those plants more susceptible to insect attack. So he's found that scale in mealybug, and quite often probably aphids as well, can be correlated with low magnesium, low sulfur, low molybdenum, low boron, low sugars or the total soluble solids, and also low manganese. And you'll see there that I've coloured those nutrients in purple, so you can see how this relates to something called the plant health pyramid, which I'll talk about in a moment. You can also see there that high nitrates are also correlated with these sap-sucking insects, so a very simple form of nitrogen. So what we want to do is create the environment where we can convert simple forms of nitrogen to longer molecular forms to complete proteins, which are in a plant available form. And the insects lose interest in feeding on plants which have complete proteins very quickly as well. So a lot of this work comes back to thinking about the plants and disease being dis-ease within the system. There's been a lot of work done in the past by Francis Chaboseau in the mid-80s, where he championed something called the triphobiosis theory, where a pest starves on a healthy plant. And the relationship between plant and parasite is primarily nutritional. And also Emeritus Professor Dr. William Albright, known for his work in soils, also talks about the fact that insects are nature's garbage collectors and disease are her cleanup crew. So if we're not producing objectively healthy plants which are fit for human consumption, we tend to find that they're more susceptible to insects and also pests, diseases. So to measure bricks, it's important to consider, I guess, some different protocols. You want to be able to take a sample from a leaf blade, the first fully mature leaf, so it might be at about node 4 or 5, and you want to be able to extract the sap from that leaf. So you need to break the capillaries in the leaf. If you're doing broadacre crops, you might be able to use a garlic press. It doesn't work so well in grapevines. We tend to need to use a pair of modified vice grips. So if you can weld, you can certainly make a pair of these yourself. And we need at least 1 or 2 leaves to be able to get 2 or 3 drops of sap out. But of course, we want to take a broader, more representative sample from the focal crop. In this case, it's grapevines. We also want to see a fuzzy line, and that is an indication that we have good levels, I think, of calcium. And we can then put those drops of sap onto the refractometer. It is refracted by the light, and we can then take our bricks measurement. So this again is work that's championed by Thomas Dijkstra, and he talks about this threshold or range of bricks where if we have a very low level below 3 or 4, then the plants are really in survival mode and they need a lot of inputs to be able to be maintained. So a lot of nutritional and water inputs. When you start getting to around probably about 6 to 8 bricks, this is when the plants can really produce good levels of secondary metabolites. So it might be anthocyanins instead in terms of colour production, and aroma as well. So if you pick up, say, a tomato in the grocery store, you'll start to smell and taste, those secondary metabolites exhibited in flavour and aroma. When you start to kind of get above 9, the plant has an osmotic relationship and it tends to have better water retention abilities, so it becomes more drought hardy. And when you get above around 14, then we can say that the plants are more objectively healthy. They're likely to have less insects and less disease. So I talked about this about a year ago to some grape growers in or through Australia actually, but some of our growers started taking bricks measurements in the Clare Valley. And you can see also here that there's the types of bricks levels where the aphid groups start dropping off, the sap-sucking insects, the chewing insects and grasshoppers and so forth. So this will be different for different crops, but it gives you the opportunity to take a benchmark and to start to observe some of these relationships if you aren't already. This is more commonly done in broadacre than it is in production horticulture, so it's great to see that shift. So one of our eco growers at Skilligalee went and benchmarked their bricks levels, and they had a pretty broad range from 10 to about 21 bricks with an average of 14 across all their production system. production units, sorry. And I asked them if there was any correlations or observations that they could make. So they said, yes, the 19 bricks reading actually correlated with the Eco Vineyards patch, where we'd converted from volunteer weed growth to a multi-species mix of diverse natives and wallaby grasses, the perennial grasses. So that was great to see this level of bricks so quickly after they'd changed practices 12 to 18 months previously. And block D15, which had a level of 21, was their top quality Shiraz, and they'd been putting lots of soil prep sprays on. So, fish hydrolysate, humic acid, seaweed, and so forth. And then this year, they excitedly sent their results through, and you can see here that they've actually had a range from 14 to 21, and they've had an average of 17 bricks. So they've had a lot more blocks transition to a higher level. They're at 19 or above bricks, which is great to see. Okay, I'd like to introduce what I think is one of the most important diagrams I've seen in my horticultural career over the last 30 years. And it's work championed and developed by John Kempf, who may be familiar to some of you from Advancing Eco Agriculture. So this is entirely his work, but it's too important not to share. And there's some great resources that have been developed on the Advancing Eco Agriculture website that you can look at to find out a lot more information. And John's also put together a series of supporting materials, so a PDF, background materials, podcasts, and so forth. So I don't tend to talk about the word sustainable. The root word of sustainable is to sustain, and I think that's too low a bar. But if we're looking for a definition of sustainable, then this is probably a good place to start. So John's an Amish grower. He has this amazing company which is consulting to about 10,000 different clients in the US, managing around about 2 million acres. And he's also focusing across at least 60 different crops. So I'd have to say that John's probably one of the most brilliant thinkers in the field of regenerative agriculture. And he's surrounded himself with mentors like Don Huber, and we've mentioned Olivia Hassan, and also Thomas Dykstra and many others. And he's distilled his knowledge and experience and vast capacity to read peer-reviewed journals, along with the application of this knowledge. So we're actually seeing this working at scale internationally, which is pretty amazing. So this concept's called the plant health pyramid. It has 4 levels, and you need to go through each level from 1 to 4. You can't just skip from level 3 to 4 per se. Level 1 and 2 talks about the plant's passive immunity capacity, and it's based on chemistry and nutritional inputs, foliar applications predominantly. And then level 3 and 4 is more around the plant has active immunity, and it can really start to produce its own defence mechanisms. So the power of this information is that while they're very complex concepts, there's some simple messages here that growers can incorporate into their normal fungicide application schedule. And as they start to see benefits, then they might be able to then pull back on their synthetic input use. So at level 1, this is all around how we can really maximise that capturing of light energy via photosynthesis. Many of our crops are probably working at less than 50% of their full potential, and some might only be working at 30% of their full potential at the moment. In order for photosynthesis to occur, you need to have a whole range of co-elicitors and or the right nutrients in balance. So it doesn't mean adding more, it means making sure that you've got the right balance of nutrients at any one time. And in the case of photosynthesis, we need magnesium and iron in particular for the production of chlorophyll, manganese is needed to split the water molecule, so it's needed for the process, but it's not an integral core of actual photosynthesis. And you also need to have some of the macronutrients in the right form at the right time, so nitrogen and also phosphorus. So if any one of these nutrients is lacking or out of balance, then it means that you're not going to have the optimum conditions for photosynthesis to occur. And of course, you need to have access to sunlight energy and carbon dioxide that can be created into air and so forth as well. So yeah, being able to maximise the photosynthetic capacity of the plants is a really important first step, and it's the basis of the plant health pyramid. And we can objectively measure this through bricks and or total soluble solids. And, you know, we need to be able to maximise the plant's capacity to produce photosynthates, to feed soil microbes, and they in turn can make nutrients in a more plant available form. And it also drives the capacity of these plants to be able to have relationships with the mycorrhizal fungi that can help produce this substrate called glomelin and maximise soil organic matter and carbon production. So yes, these are some of the nutrients needed to maximise the photosynthetic process. At level 2, this is all around the use of nitrogen in its different forms and how you can convert very simple forms of nitrogen, so like nitrates or ammonium, through to complete proteins. And in order for this to occur, again, we need to have the right balance of nutrients applied as foliar nutrients. So we need to have magnesium, sulfur, molybdenum, and boron. It's also important to remember that you might have sufficient levels of these nutrients in the soil, but if they're not in a plant available form, then the plant isn't able to access them. So if you've got a high pH or an alkaline soil, then you might get magnesium and iron bound up. So you need to consider perhaps applying chelated forms so that they're in a plant available form. And as we do this, we find a few things occur. We find that as we're able to move from simple forms of nitrogen to complete protein synthesis, that insects lose interest in feeding on these plants. But we also find that simple forms of nitrogen, say nitrate, can actually create a big yield lag on the production of the plant. It might be a 20 or 30% lag because so much energy is required for the plant to actually convert the simple forms of nitrogen into complete proteins. And there's water required in order for this process to occur as well. So you may actually have an extra burden of the plant requiring, 20 or 30% more water in order to be able to convert simple forms of nitrogen into complete proteins. So yeah, I think it's a really interesting line of inquiry, and I'm interested in the soldier fly frass production, because that's more of a complete protein. So yeah, it's a really fascinating, I guess, concept. So that's what happens at level 1 and 2. And most of our plants at the moment are probably sitting below level 1 in terms of their objective health. So if we can get that balanced out, and it might mean taking leaf blade analysis and sending them off for sap analysis of a broad range of nutrients, that's important to take those samples before 9 a.m. And to send them off to a lab that can give you the oversight for a complete nutrient panel, so that you can make objective decisions about what's required. And then level 3 and 4, I guess, is almost a step change. And a lot of the requirements here come back to the plant's relationship with the soil microbes and an abundance and diversity of a soil microbe population. So that's required for the plants to be able to produce adequate levels of the photosynthates, and when they have excessive photosynthates, they're a bit like us, they store them as fats, or in the case of plants, it's lipids. And this is like when you see the outside of a leaf and it's got a very shiny, waxy kind of coverage. we know that the plant is then able to produce these lipids, and that's the plant's natural defence system against fungal pathogens in particular. So every time we spray a penetrant, we're actually breaking through the plant's natural defence system, which is a bit counterintuitive. The other thing I didn't mention is at level 1, where we have complete photosynthesis occurring, we tend to have less issues with soil-borne pathogens. At level 2, where we have complete protein synthesis, we tend to have a lot of the sap-sucking insects, kind of lose interest in those plants. Level 3 is where we start to get resistance against fungal pathogens, powdery downy, mildew, botrytis and other bunch rots. And level 4, again, is reliant on this relationship with soil microbes, and they're having everything in balance at level 1, 2 and 3. And this is where the plants are able to produce their own phytonutrients. So this is in the forms of, say, jasmonic or salicylic acid, where they're able to produce their own defence mechanisms, which help, again, against a lot of these insects and diseases. And it's at this level that we start to see reported levels of, if you have nematodes, they're less of an issue. You might have some viral pathogens that may still be there, but they exhibit in a less damaging form. So it's really interesting to kind of think of this hierarchy. And if you can get up to level 4, then the system really starts to become able to self-heal to some degree, but you should be able to then pull back on a lot of the synthetic inputs that are currently going into that system. It's grounded in some amazing peer-reviewed science, and also the demonstration of that science in real-world examples. So I'd encourage you to find out more, and there's a lot more information and detail that goes into that particular concept. So I just wanted to make you aware of the plant health pyramid and how powerful that potential concept is in terms of us being able to create step change in the way in which we manage some of our pests and diseases and create objectively healthy focal plants. The other really exciting aspect is that technology is now catching up, and I don't tend to be a big fan of technology. It quite often overpromises and underdelivers, but this particular bit of ag tech I've been really impressed with. It's called Nutritech by Sensine, and they've worked very closely with Olivia Hassan, who has been working with Redox to put together a gadget that you can use in real time in the field, so that you're not having to worry about the messiness of doing bricks or sending samples off to the lab. You can actually get, I think there's about 12 different nutrients or 13 different nutrients, with a couple more coming online, hopefully in the next 6 months in the northern European growing season. Which allows us to be able to assess the nutrients in real time in the field, and also to look at the redox, the pH and the bricks, and a measurement of EC as well. I'm not sure you can see chlorophyll with grapevines, but you can with some of the other crops. So they've got some really good data sets that they've pulled from Nova Crop Control in the Netherlands for probably about 6 different crops, some broadacre, grapevines, tomatoes, and a few others as well. So I've got one of these gadgets, and I'm just starting to trial it out, and also then look at comparison results from the lab. But it shows, I think, tremendous promise, and there's new gadgets on the way to look at objectively measuring soil health and liquids in the future as well. So I'm really excited by some of these new developments. Technology is really catching up, and this is an example, I think, that is working really well. So I'd love to invite you to join the Eco Vineyards community, or Eco Grower community, as we'll talk about more so going forward, so that it's not just focused on wine grapes, but we're working cross-sectorally across a whole range of different crops. If this is of interest to you, please get in touch with me if you'd like to find out more. We have a range of grower case studies and insights. We've got 76 demonstration sites set up across Australia, 67 case studies. So growers have been putting this information into practice and they've all approached it from a slightly different perspective. So there's some great insights there. We have a whole range of different case studies. This is one from the Mornington Peninsula that Karen's been inherently involved with, looking at using pheromone as a mating disruptor, or as they say in France, creating sexual confusion for our tortricid or our light brown apple moth pest species, which has worked really well to reduce damage to the developing inflorescences and therefore botrytis and other bunch rots. As I mentioned earlier, we've got lots of resources available. So we have the best practice management guides, but we've also got more than 2,000 pages of practical resources for wine growers. And if you'd like to read a hard copy, you can go onto the Eco Vineyards website and order hard copies to be sent out as well. So if you'd like to search information, you can do that by topic, by resource type and by region. And we also have a regular newsletter. If you'd like to sign up to that, then please zap that QR code. Or you can do that by filling in your details on the front page of the Eco Vineyards website, right at the bottom of the page. We also have a calendar which helps guide growers with the different practices throughout each month of the year. And we'll be developing and launching a podcast soon with myself and Dan Falkenberg, who's a multi-generational viticulturalist and award winner from Eden Valley in the Barossa. But until then, I'd encourage you to check out John Kemp's Regenerative Agriculture podcast. He talks with some of the most brilliant minds in this field, and it's a fascinating listen if you're spending quality time out on the tractor. So you can contact me via retallack.com.au or via the ecovineyards.com.au websites. And thank you for your time today. I think there might have just been a couple of questions. I've probably covered most of the ones that I've seen, but there was a couple more that I can just touch on briefly. So one of the questions was how to measure or to be able to develop these carbon properties in soil. Just to give you one example from Dan Falkenberg's property at Edenhull in the Barossa wine region, he's been growing a diverse mix of native grasses and forbs, including wallaby grasses in the mid-row and also the undervine area. And they've been under drought for a number of years. And since 2021, where they started benchmarking their carbon, it was 1.2%. They benchmarked it earlier this year and it was 5.9%. So they've had an increase of nearly 1% of soil organic carbon each year over the last 5 years. And they've also found that their light, sandy, acidic soil with an EC of 0.3 has actually halved and it's down to 0.16. So they've seen lots of benefits there in terms of their soil quality and water holding capacity as well. So that's just one example, but it comes back to, again, having 100% ground cover and functional root growth wherever you can, 100% of the time, wherever possible. And another question, I think, was building farm fertility using on-farm generated inputs without the need to apply toxic pesticides or synthetic fertilisers. So yes, if you can cover your soils and nourish your soil microbes, they will then start to be able to produce, if the nutrients are there, the nutrients in a plant available form. So you can nourish the soils by putting on inputs like fish hydrolysate is really good for more fungally dominated soils, along with humic acids, seaweed extract, and that type of input, a little bit of molasses perhaps. And some of the take-home messages I won't go through in a lot of detail, but they're here if you'd like to have a bit more of a read. We really try and work from a position of kindness and generosity and cultivate a polyculture of thought, to take an ecology analogy. So, we're looking for lots of input and grower experiences. We've all got a little bit of the picture, and, if we can pull all of that information together, we all benefit. So I encourage you to be kind and generous with your knowledge. That sense of reciprocity in building trust means that we all get to learn so much more quickly. As I mentioned, we need to cover and nourish our soils and to feed your soil livestock, so that's the microbes. We want to incorporate functional ground covers and have active root growth 100% of the time wherever possible. In terms of ground covers, we want to move from a monoculture of a single species to a polyculture of multiple species and from different functional groups. And if you look at the best practice management guide on soil health, ground covers, we have lots of insights to the different types of cover crops, but also combinations of different native grasses and forbs. We want to move from annuals to perennial ground covers, so we have more fungally dominated soils, especially in perennial production systems. We want to move from just grasses to actually a diversity of flowering plants or forbs, ideally a mixture of both. And we want to try and move from exotic to more native or locally adapted species and incorporate agroforestry where appropriate. We do this using insectary shrubs for habitat and also IPM benefits. So all of these are actually related directly or indirectly to pest and disease management. We want to maximise the functionality of the system and the life both above and below ground. And yes, scruffy is perhaps more functional. It's the new look. Make sure that your soil can breathe. So we need to address compaction issues. That also relates to redox. Be kind to earthworms. We need to really address, these soil compaction issues. It's something that quite often goes unaddressed. So please check out the work of John Kemp and that of Advancing Eco-Agriculture around nutrient integrity in the plant health pyramid. And I'm all for ripping off the Band-Aids, so breaking that cycle of intervention and embedding longer term solutions. And just to finish, it's important to remember that we are nature, and what we do to nature, we do to ourselves. So let's nourish not only individuals, but our production systems. And in doing so, we're also nourishing our communities. So thanks for your time today, everyone.
Awesome. I'm just going to thank Mary for her presentation, and thank you for her for putting the time into doing a recording for us. Any questions, please put them into the Q&A. We will organise for Mary to answer any other questions into the new year. I'd like to hand over now to Joel. And Joel is going to finish with his presentations on plant nutrition as well. And then we will, if we've got time, go into some live questions with Joel at the end. So over to you, Joel, to share your presentation. Thank you.
Awesome. Thank you very much, Karen. Okay, can everybody, can you see my, hopefully my screen there?
Yep, looks good.
Okay, thank you very much. Okay, so let's dive into it. So I'm going to touch on a couple of areas that are complementary to what Mary has outlined, and also some additional aspects. We'll go a little bit into that nutrition, also complementary, and a little bit into the biology as well as another piece that we'll cover. So my framework at looking at kind of plant health and pest and disease and minimizing pest and disease resistance predominantly looks like this. I look at this point of saying, let's modify the environment. Let's create the environment that is less conducive to pest and disease. And actually, I think this is actually a fundamental first step. We have to be creating environments that are less conducive, because then as a secondary, our integrated pest management strategies, be that all of the tools in your toolbox that you're going to integrate, that they're all going to work better, more effectively, where the environment is creates less pressure in the first place. So here we're going to dive a little bit into some of the biologicals, microbials that can help with pest and disease, and we'll expand in the nutrition, like Mary has just touched on as well. And my point being is there that often there's a bit of a healthy skepticism around the potential of biology or nutrition to control pest and disease. And I think that it's a fair comment that they are not perfect tools. I think that all tools are a little bit imperfect, and that's why we have integrated management approaches. That's why we bring many together. And my point here is just to say that where biology and nutrition are hugely helpful tools, they work better in an environment that in which they will function better. If we have not created the right environment in the first place, we'll find that these tools are a little less perfect. And that's kind of what I want to touch on. Okay, so let's dive into the environment. And the point here about modifying the environment, there's actually many aspects to this, but the one key point that I want to iterate in this point is just diversity. It's just to say that we want more diversity in our production systems. And look, I know there's some we've got a few mixed farming operations here in the room today, and who'll tune into the recording, but diversity can mean diversity through time. And like that's a temporal diversity, that's rotation, crop rotation. So that is an example of diversity through time. But we can also add additional layers to that diversity. And that could mean spatial diversity, where we are physically separating different plant species or different productive areas, for example. An example like this would be the vineyard and the interrow, like we've just heard about, but it could also be tree crops, trees and cropping. It could also be even just an intercrop, an alternate row companion or intercrop, for example, a piola, pea and canola. So we are spatially separating the two plant species is the point. It's a spatial diversity. And then the next kind of dimension to diversity is genetic diversity. And this is really the idea here is varietal blends or blending cultivars. And this has a lot of opportunity in that, sure, it's still a monoculture of wheat or canola of whatever it might be, but by blending different varieties together, we create a better depth of the genetic diversity. And this also adds to the pest and disease resistance, because some of those varieties in that blend are going to be resistant, more or less resistant. And that's kind of the idea here. When you plant a monoculture that is a single species, that is the same variety of the same species, it is a absolute monoculture whereby once one plant is susceptible to that pest or disease, all plants are susceptible. And that's kind of the weakness. Of course, monocultures have a lot of strength about them in terms of their ease of sowing, implementation timings, harvesting. Like there's many aspects to a monoculture that are advantageous, but their inherent design flaw, the weakness of them, is also that they are all uniform, that they are all the same, and therefore they are all susceptible to that pest and disease. And even a bit of genetic diversity through variety blends, blending different varieties together, can also bring a lot of advantages. So here's just a small example of this. Here you're looking at a 7-way wheat blend in this example. And look, a lot of farmers are doing more like 3 or 4-way blends. But this is an example of a 7-way, but there are also examples even higher also. Anyway, my point being, sure, you're still looking at a monoculture of wheat here. You can still seed that at the same time, harvest that at the same time, etc. But you've just got a little bit of genetic diversity, and that the biggest benefit to this genetic diversity is lower pest and disease pressure. And I wouldn't say always, but often, but I wouldn't say always, but often it does translate, therefore, into yield gain. But I wouldn't say that's a guaranteed from what farmers have kind of fed back to me. But certainly it can be in many instances. Okay, so here's an example of spatial diversity. We are physically separating, this is pea and mustard as an example, alternate row intercropping. We're going to crop these together. We have these alternate rows of pea and mustard. And as an example, just think about this. If a disease spore is blowing into this field, by design of the production system, by modifying the environment, now as those spores blow into this environment, they are only hitting a host plant 50% of the time. If it's a disease that affects the legumes, for example, okay, it's hitting all this mustard, right? Okay, and so the spore is not going to germinate on the mustard. It's going to germinate on the legume. Okay, so by default, we have just lowered the spore load. We've lowered the disease pressure by 50 odd % just because half the spores are landing on non-hosts. And then when those spores, however, when they do germinate and do start to spread, okay, now they're in here spreading. As those spores pop and mature and spread into the wind, for example, okay, now boom, we are hitting a barrier. We're hitting a non-host again of these mustards, brassicas in this example, where we now, again, we're spreading, but we're controlling that spread through this diversity, through non-hosts is the key point. So there's a nice example of spatial diversity. And here we have just one of the classics idea of kind of the polyculture or the plant species diversity over on the right-hand side here that you can see. And the point here is, of course, as I'll also show on my next slide, is just that when we dilute, see this monoculture here, same plant species, same variety, when we dilute this with non-susceptible species, with non-hosts to the pest or the disease, now it's harder for that disease to spread through this canopy, spread through this environment, because we have all these non-hosts in the way, and that is going to slow the spread. And that is the key kind of point. So with more diversity, we dilute these particular hosts of interest, and we put all these non-hosts in there, and that creates an environment that is not so susceptible. And this slide here is exactly supporting this point. You're looking at increasing plant species diversity and how that impacts on pathogen diversity. And so you can see that as we add more hosts, as we increase the plant species diversity from 1, 2, 4, 8, 16, for example, that we are actually increasing the diversity of the number of pathogens, or as the plant species increases, we increase the number of pathogens who are present, who we will find in that environment, which of course makes sense. It's because we have more hosts, different host plants, we have more diversity of pathogens that can cause disease on those hosts. So that makes a lot of sense. However, the key point here is that what about their ability to cause then disease, to cause infection, their overall infectivity? And as you can see on the right-hand side, as we increase plant species diversity, we actually are lowering or reducing the infection, the ability to cause disease, and simply again through host dilution. As we have 16 plant species, for example, now the pathogen as it comes in, or the insect, it's now going to face 15 other different plants before it can find the one again that it's interested in. So we've mixed it all up with these mixed signals, 15 other plants before we can get to that 16th one, which is the same, and then cause the problem. Okay, so overall, your ability to cause pest and disease pressure reduces as we increase the plant species diversity. And that's where, if we can just modify the environment to begin with, instead of vast areas of monoculture, which are all the same and all susceptible, if we can just recreate the environment to integrate some of these kind of, rotations, diversity through time, some of the examples we talked about there, intercropping, companion cropping, but also some of these areas here, the non-productive areas. What are we doing with our shelter belts? What are the opportunities around some of the low-lying areas of the field, buffer zones, pollinator strips, inter-row areas, again, in Mary's examples there of the vineyard or an orchard, tree crops, for example. What are we doing with the inter-row areas and these non-productive areas? Again, also buffer zones, field margins, and the opportunities to create biodiversity hotspots there with these flower strips, bird seed mixes, et cetera, which create habitat for wildlife who then migrate into the productive areas and provide pest and disease resistance, pest and disease services, controlling pests, controlling disease as they migrate, as those beneficials migrate into the productive areas. So, okay, there's just a few examples. It could be, alternate row intercropping. This is a chickpea flax example here, diverse cocktail cover crops, you name it. So it's basically just about bringing a range of different plant species and varieties in different capacities, be that within productive areas, but also outside of the productive areas in those non-productive areas, and really maximizing the diversity in both of these areas at a field scale and a landscape scale, to create plant diversity, to create diversity of insects, of beneficials, of birds, of wildlife, of ecology, which Mary outlined some wonderful examples there in her lecture there. And they will provide the service. They will provide the pest control service if we provide the habitat for them. Okay, so that's environment. Let's modify the environment. And the key message there is more plant species diversity. Okay, redesign with diversity to modify the environment, make the environment less conducive. So then we move on to biology. Now, when we talk about biologicals and nutrition in order to try and control pest and disease, we're going to do that in an environment that is more biologically diverse and therefore harder for that pest and pathogen to be causing problems. And that's kind of that core idea of the environment there first. So you've all heard of the disease triangle before as an example, and it's a common thing where we talk about, oh, in order for disease outbreak, you need to have the host plant needs to be present, the pathogen needs to be present, and the environment needs to be optimized, suitable, adequate, for the pathogen. And when these three things align, when they all line up, then boom, we have the opportunity for disease outbreak. And this is this classic idea called the disease triangle. I'm sure many of you have come across this idea. I just want to point out what is absolutely perfectly good framework to think about pest and disease, but just pointing out one of the obvious and massive oversights of this, which is the role of the beneficial organisms, the soil microbiome, for example. These beneficial microbes, they can suppress the pathogens, eat them, antagonize them. They can enhance plant immunity. They can change the environment, reaggregate the soil, create the soil better structure for more oxygen, etc. They do a lot. They interact with all of these three aspects to the disease triangle. And yet in classic disease triangle teachings, it is completely neglected. So point being, the beneficials are a huge opportunity here to tilt the balance of this disease triangle towards better health outcomes. So for example, okay, yes, biology can control pest and disease through multiple ways, through exclusion for space, so competition for space, nutrients, and the like, through antagonism, okay, so amongst the microorganisms anyway, we're talking here about producing a bunch of antifungal, antibiotic type compounds, so various microbial metabolites that are inhibitory in their mode of action. Predation, and this is true then, of course, especially amongst the insects. We have all sorts of beneficial predatory insects, and we also have predatory fungus and predatory microorganisms who eat pathogens, for example. So this is another aspect here, and as well as then enhancing the plant's own immune system is an extra layer of defense. So here's just one small example of this, pointing out that no rest for resting spores, that there are many beneficials who eat disease spores. The spores in which diseases, of course, germinate from and grow and spread, those are food sources for many other beneficials. And here's just some examples whereby there are, in one example of clubroot disease in brassicas, we have actually many identified predators, things that eat those spores before they've even germinated. But the only way you're going to get these beneficials to eat those spores is to have good soil health and good soil function in the first place, so that you have a good population of these beneficials who are going to do that job for you. So that's just one small example of predation. Many of you, I'm sure, have come across this idea of compost extracts, this idea of mixing water with compost and washing out some of the organisms out of that compost, particularly the microorganisms, and then applying those to the seed or injection into the furrow or seedling treatment or foliar spray. as an examples, but and just to point out that when you inoculate these beneficial organisms, they have various ways and means in which they suppress and control pests and disease. And just to give you an example amongst some of the major diseases, if you look into the academic literature on this, I'm not saying that compost extracts or compost teas, that they are a foolproof measure, the silver bullet, and that they always control pest and disease. Sure, they can do in some instances, but more so what I'm pointing out is that they are a tool in the toolbox. They are part of integrated pest management. And look, if you look into the literature, yes, we have evidence that they can control, that they can suppress many of the major plant diseases. Now, okay, can they control and do that every time, all the time, in every environment, in every year? Okay, no, probably not. But can they be a piece of the puzzle that can help as part of an integrated approach? Then yes, they can provide a certain be part of that package, that integrated pest management package. And yeah, again, if you look at those major pest diseases listed here on the screen, look, we have evidence that compost extracts have at least some suppressive qualities, properties over these disease organisms, for example. So look, I think my point is just say that they can be a useful piece of the puzzle, and really in terms of our understanding about the microbiome and its role in managing pest and disease, that it's a very, very complex world, and actually we know very little about it. And so at the moment, one of the best strategies, a bit more of a scattergun approach in order to manage pest and disease, is to just encourage microbial diversity. The more microbial diversity, the more likely you are to have the organism present who's going to control that pest or disease. And so just as an example here, this is a recent review on this, pointing out that the soil microbiome is a critical component of healthy soils, and that increasing evidence suggests that soils with low diversity or homogenized microbial systems, so very much more narrow, dampened diversity, that these are more susceptible to soil pathogen invasion. But the extent and mechanisms that increase the threat of pathogen invasion, such as increasing the prevalence of the existing species and introducing those new species, remain unclear. So what we're pointing out here is just that there's a lot of knowledge gaps, but what seems to be the case is that the greater the microbial diversity, the more likely we are that some of that diversity is going to, in various ways and means, is going to control the pest and disease as it tries to colonize that new environment. Okay, so that's just a few examples around biology. Let's move on to talk a little bit about nutrition as our third piece of the puzzle, and then we'll wrap it up with some chat and discussion. So Mary touched on this a little bit earlier with the kind of overlapping a little bit here with the soil health, with the plant health pyramid, this idea of essential nutrients, and they are the keys, micro minerals. They are the keys to good plant growth and productivity, but they are also the keys to good plant health. And so when we talk about photosynthesis, look, it's not just about the sun's energy, it's of CO2, it's water. Yes, those are fundamental, but it's also the role of nutrition, essential macro and micro minerals, which help to make this process happen, to help to support photosynthesis and drive, be that productivity, but also be that drive the production of some of the plant health, plant defense chemicals and plant health compounds that help it to resist pests and disease as well. So nutrients, they are the things that catalyze photosynthesis, but then those products of photosynthesis, the sugars, carbohydrates, etc., those are then turned into defense chemicals. That's through kind of some of that secondary metabolism. And essentially nutrients, micro minerals, are the keys to driving photosynthesis, but also driving secondary metabolism. They are the catalysts that make all of this happen in order to help the plant build its defense chemicals. So this slide here is really what we're emphasizing it's the process of photosynthesis, CO2 plus water in the presence of sunlight, but it is the essential microminerals, which are formed part as enzymes that catalyze the reaction that help to form sugar or glucose plus oxygen. Then that secondary processes, that sugar, the product of photosynthesis, is then turned into all of these other things, all of the things that the plants are made up of, everything that the plant requires, that it is made up of, come from this sugar, which come from this carbon, which was out of the air. Okay, but again, in order to fulfill the second step, it is essential microminerals, which also catalyze this step to build everything that the plant is made up of. Complex sugars, carbos, amino acids, proteins, the lipids, hormones, vitamins, smells, aromatic compounds, but particularly here, these defense chemicals, some of these protective and defense chemicals. These are the ways in which the plant defends itself. And these compounds need nutrition to make them. And they're made from the carbon, from the sugar, or the backbone or the skeleton of that carbon is used to build some of these other defense chemicals. So let's take a quick dive in our last bit of remaining time. I'm keeping my close eye on the time. We're going to take a quick dive into the role of some of the nutrients in supporting this whole process. So just so we're clear, there's actually a lot of literature and evidence to support this idea that plants that have nutritional deficiencies are more prone to pest and disease attack. It makes them weak. They are weakened without the essential nutrients that help to catalyze the production of the defense chemical or other compounds. And without that nutrient, the plants become weakened, and therefore pest and disease can exploit that. So just so you're clear, it was just so we're all clear, there's a lot of actually evidence to support this, and so much so that you've got reviews, papers that just review all of the other evidence that go through this. And I'll come to the real challenge in this discussion in just a minute in a future slide. But just to kind of set the scene here, I'm just pointing out that, look, there is a lot of evidence that supports this idea that nutritionally deficient plants are more susceptible. And this is one of those, a review role of nutrients and controlling plant diseases. Here we are pointing out that when there's high nitrogen, there's an increase in severity of infection, that potassium decreases the susceptibility of the host. Manganese can control a number of diseases through its role in lignin biosynthesis, which is structural barriers. Phenol biosynthesis, that's some of the defense chemicals. And as Mary mentioned, also in its role in photosynthesis. Boron here also found to reduce the severity of many diseases, because boron is very important for cell wall structure. It makes the cell walls very strong, reinforces them. And along with silicon, which also does the same thing, shown to control a number of diseases because it helps to create this physical barrier. So it's calcium, silicon, and boron that all do this, that can also particularly restrict fungal penetration. But also silicon is important for the production of defense chemicals. So how does the plant defend itself? One, it's through physical defenses, the barrier between the outside environment and the plant itself. So here we're talking about in this epidermal layers here, with optimized nutrition, the plant can produce lots of cellulose, it can produce lots of pectin, it can produce lots of lignin. So it's structurally tougher and more rigid in that membrane area. And then we have these cell walls that you can see here, and that's the calcium, silicon, and boron example. They deposit particularly right here in these cell walls, making those very, very tough and rigid. And essentially that makes it harder for pest and disease to penetrate, to chew through and cause damage. Now, we also have internal secondary defenses or biochemical defenses, and these are the systemic chemicals that plants produce internally. And the key point about these is that when the plant is under attack, be it from a microbe or an insect, the plant detects that and it sends signals down to the roots where it changes its root exudation profile. It will release stress chemicals, and it will release these to recruit the armory. It will recruit beneficial microbes to help it mount its defense systems. So the trigger is the pathogen or insect attack. The plants release these root exudates to recruit or wake up many beneficial microbes who answer the call and who will crowd around and grow amongst that root system, where those organisms will help the plant with its own immunity. They will help the plant mount its defense chemicals, and it will translocate those defense chemicals throughout the plant, whereby the new leaves then will be protected. So all I'm pointing out here is that this biochemical defense particularly is actually a really nice example of both biology and chemistry, both biology and nutrition, because it is the plant waking up the biology. But when the biology are recruited and help the plant make those defense chemicals, the plant still needs nutrition, micro minerals to build those defense chemicals. And that's where the biology is important to supply those nutrients to help induce these signals and drive some of these benefits. So I would summarize the nutritional aspects, the key points here in terms of the key pest and disease fighting nutrients. I would focus here on calcium, silicon, and boron. They're in the cell walls there, as I mentioned. So I would be proactive about these three nutrients. Manganese, copper, and again, boron. They're also really important for lignin, cellulose, pectin synthesis. So that's those other structural compounds. So for physical protection, it's calcium, silicon, boron, manganese, and copper. When we talk about some of the internal systemic chemicals, defense chemicals, silicon is again very important, manganese is again very important, and sulfur is also a very important nutrient for some of the defense chemicals. And then I would just also emphasize the role of nitrogen or imbalanced nitrogen, excess nitrogen. If we have too much nitrogen in the system, in the plant particularly, this makes the plant more attractive to pests and disease. And I will quickly outline the whys of that super quickly now without this last few minutes here. So Mary mentioned the French agronomist there, Chaboseau. He put this idea many years ago around that nitrogen imbalance creates this plant, poor protein synthesis, and a lack of nitrogen imbalance in protein synthesis makes the plant more attractive to pest and disease. And this slide you're looking at here is a very recent review of this idea. So putting some academic kind of published evidence behind this idea, saying, well, does excess nitrogen or imbalanced nitrogen create pest and disease problems? And they bring together, in this review paper, they bring together the evidence that supports this idea and highlights that when we have high nitrogen, they use the word high nitrogen. I would use the word imbalanced nitrogen, but it's a subtlety. But they point out here that high nitrogen leads to altered defense chemicals. And as I'll point out in my next slide, that can make the plant potentially more attractive to pest and disease. But the key point here is that it leads to, if we don't have adequate nutrition, optimized balanced nutrition, it can lead to an accumulation of free amino acids. And this is what kind of particularly Mary mentioned the nitrates, high nitrates, that sap-sucking insects are quite attracted to the high nitrates. But all the insects, they're quite interested in the amino acids, because amino acids are the building blocks of their bodies, just like they are owls and plants and microorganisms and livestock and chickens and everything. So plants that have lots of these become more attractive to the insect pests. But when we stitch those amino acids together to form proteins, full, complete, and complex proteins, now this becomes less attractive to the pest and disease. And that's why that important emphasis in the plant health pyramid is on protein synthesis. Once we build proteins, we stitch those free amino acids together into proteins. Okay, now this becomes less attractive to the pests, insect pests, because they can't digest those proteins. So that's kind of the core of the idea. And if anyone's interested, you can Google this paper and have a read through this one. They also highlight that when we apply pesticides, that there is a stress response in the plant, in the cash crop, and actually this leads to an accumulation of amino acids. The stress response, it leads to protein breakdown, which leads to an increase in amino acids, which makes the plant more attractive to pests and disease. Now, super quickly, because I'm running out of time now, so I'll just emphasize nitrogen. When we have high nitrogen, that can increase the nitrogen containing defense chemicals, and that can be a good thing. So if the insect pest is susceptible to the nitrogen bearing defense chemical, that would be a good thing. If we have high nitrate, we see high nitrogen bearing defense chemicals. But there are another group of defense chemicals which are more of the carbon bearing, and they are suppressed by high nitrogen. So when we have high nitrogen, we decrease or suppress these carbon containing defense chemicals. And they are, if your insect pest is susceptible to those, then that's going to be particularly problematic. Okay, so this is where high nitrogen imbalance can then cause particular problems. And this was my point I was just trying to make earlier about if we have an accumulation of amino acids, that makes the plant more attractive to pest and disease. And that's particularly if we're lacking some of these minerals here, like phosphorus, sulfur, magnesium, manganese, boron, potassium, zinc, copper. All of these nutrients are involved in good protein synthesis. And so if we are lacking these nutrients, amino acids begin to accumulate. And that's where the plants become more attractive to insect pests, for example. And Mary mentioned that example of high nitrates. It's absolutely spot on as well. If you have a molybdenum deficiency, a sulfur deficiency, an iron deficiency, well, then that nitrate starts to accumulate and bottleneck, and it can't move down this metabolic pathway. So it doesn't matter what form of nitrogen we apply, whether it's nitrate, urea, or ammonium, they all want to be converted into amino acids and then into proteins. But in order for them to do that, they need these key nutrients here in the red boxes in order to support each step along the way. So I will just point out here that the evidence about plant nutrition helping to reduce pest and disease pressure is plentiful in the academic literature. That's not the problem we have. The problem we have is not about the evidence to support this idea. The problem we have is putting it into practice. And here we have a really nice example of this about foliar spraying manganese on cucumbers to control mildew. And this paper kind of illustrates this really nicely in that they point out that, look, foliar applications of manganese, this is an effective factor in enhancing plant resistance against fungal diseases. But the efficiency of this method strongly depends on the application time. So what they found here is when they applied manganese, they got a good spike in manganese levels in the leaf, and this led to a significant reduction in the fungal disease. But they did this at different timings. So the highest reduction in disease severity was observed when manganese solutions were applied at 4 days before infection. So they applied the manganese treatments and then they inoculated with the pathogen. So the cucumber plants that were treated with manganese at 4 days before infection accumulated the highest level of manganese in their leaves and higher than those treated at 14, 10, or 7 days before infection. So if you applied that manganese, 2 weeks earlier or 10 days earlier or 7 days earlier before the infection, you didn't get as good control. But if you did it at the optimum at 4 days, you got the best level of control. So here's a really nice example in practical reality. How do we ever know what day, when we're going to get infected by a pathogen? No, we don't know what that day is. So that becomes the practical challenge of putting, putting this into practice. How do we know this information? How do we optimize the plant nutrient levels during these critical stressful stages. So I think this example exemplifies this really nicely. Look, we're not debating whether manganese helps here and whether manganese control disease. What we're debating is the optimum way to supply that, the optimum timing, the optimum source. That's the part that really needs to be unpacked more so, not the idea that nutrition can control disease. And this was just, I'll scoot through here because I know I'm on time now. So here I was just pointing out a nice example of foliar applications. This is an example of zinc. And you can see here that if you are trying to correct a deficiency of a nutrient that you want to, the fastest way to correct that and address that is through a foliar application. And here we're just comparing a couple of different zinc sources, and you can see on this side here compared to here where we have much vibrant zinc absorption taken up by the leaf here. So if you're trying to address this mineral factors, it's foliars is the quickest way. And I'm going to skip over these now just to wrap up because I know we have to have some questions. So all I was going to I did mention briefly silicon already, so I'll skip over this now. But just to say that, look, silicon is a really good example of a key nutrient that's very well published, very well evidenced, and it really helps to reduce pest and disease pressure. And also help with a range of environmental stresses. Here's an example, a review paper of impact of silicon-enriched plants on pest attack. You can see that anything to the left of this line is having a negative impact. So we can see it reduces fluid feeders, chewers and borers, especially these latter two. Silicon-enriched plants have less pest and disease pressure. And one of the other important things that silicon also does within the plant is it drives the production of some of the volatile organic compounds, which actually attract in the beneficials. So when the pest is attacking, and the plant can release some of these stress, I mentioned stress rutexitates, but earlier they can also release stress volatile organic compounds up into the air. And this can actually attract in the predators. And silicon-enriched plants have a higher capacity to do this. So silicon as a nutrient is a very, very important one. Okay, so in summary, and we'll wrap up here now. Look, my framework here focuses strongly on the environment first. You have to accept if you're going to plant a monoculture of the same thing, of the same plant species, of the same variety, if one plant is susceptible, they're all susceptible. So any which way that you can break up that monotony, that monoculture with diversity is going to reduce the burden, slow the spread, and reduce that pressure. So the environment, I think, is a fundamental redesign. We've got to redesign with diversity, and that's going to make the environment less conducive to pest and disease pressure. Then when we use our added layers of biology and nutrition, I think they're going to do a better job in this more diverse environment because the pest pressure is lower. So biology, that's going to mean beneficial organisms which can outcompete the pests. That might be beneficial insects, or it might be some of those beneficial microorganisms which can also produce antibiotic type substances, but also support the production of some of those defense chemicals. And then nutrition is absolutely key because the plants need the nutrients to make the defense chemical. They can't synthesize the defense chemical without the key nutrients. And that's where some of those essential micro minerals are fundamentally important. And that's where you have to take some, I think, leaf analysis or sap analysis to identify what your nutritional constraints are. And then you can address those accordingly. And as I mentioned, a foliar spray is particularly a good avenue there. Okay, so I know I went a few minutes over time also, so hopefully we've got a couple of questions for some wrap-up chat. Okay, thanks very much.
So thank you so much, Joel. Just a reminder, yes, if anybody does have a question, please put it into the Q&A. The chat has been disabled, so you'll need to use the Q&A across the top of your screen to put any questions in there. If we don't get any particular questions in a hurry, I'll pick on people. I also think that the raise the hand is there as well. So if people do want to ask Joel live a question, please do that as well, rather than put it into the chat. If that's an easier thing for you to do, just raise your hand and speak up. So I'm just going to hand it over to everybody. If anyone has any questions, raise your hand or drop it into the chat. Or I'll hand pick people. Cool. Thank you, Richie and Rosie Growing Organics, for your hand up. Yeah, if you want to ask your questions, go for it. And just remember, you might be on mute. Hoping. Oh, maybe we can't hear people. Yeah, if we can't hear people, even though you can put your hand up, you might have to put it into the chat. Sorry, the Q&A.
I have allowed mic for them. They should be able to speak now.
Okay, all right. Richie and Rosie, are you able to find the unmute button? Anybody else have any questions while we're just trying to work out if we can get them on some audio? Rhiannon Stanford's there. Rhiannon, you've always got a question.
Okay, I found it.
Oh, good. Yeah, here we go. Woohoo! You're saved, Rhiannon.
I'm used to Zoom, not this one. So I've just found the unmute button. Richie here from West Gippsland. Thanks for the wonderful presentation to both Joel and Mary. Look, have you added or included or thought of agri-solar as another contribution to your agrobiodiversity components? Because I've just read a wonderful report from Solar Europe, where they've got a report of 14 different configurations of agri-solar, from broad acres to vineyards to orchards to row crops to all different sort of configurations, and they only they had a 180% increase in yield in vineyards overall. It was like a 5% reduction in ground cover area, but something like a huge increase in actual vine yields. Biodiversity increases from almost all the measurements on agri-solar integration, solar between and above and below crops, particularly solar over 10 to 30% of the area in between the rows. Seems to increase yields, reduce extreme temperatures in hot and cold weather, increase biodiversity, as well as get income from the solar panels. So it's a biodiversity contribution is a major finding from the European study.
Wow. Yeah, very interesting. Look, I'll be honest and say I'm generally a bit lukewarm on the solar discussions. And by that I mean on good agricultural land. I do have some reservations about that. I think that we should be plastering those things all over the cities and the buildings and the infrastructure all over cities well before we even consider it in putting it on soil. But yeah, your comment is very interesting because you've highlighted a lot of co-benefits, it seems, to biodiversity and productivity. So yeah, look, I've scribbled that down. I'd love to see. I'm going to go Google that report. I'd love to see that. That's very interesting to me because I generally look. Of course, they have a role in diversity of business income and that kind of thing. But I wouldn't have ever guessed that they would increase the diversity of some of the biological aspects. So
yeah,
thank you for that.
Yeah, there's also in Australia we have a lot of broadacre sheep grazing country, and they found when you put the solar panels a metre or more above the ground rather than close to the ground, and you construct a frame so the sheep don't damage the frames when they rub against them. By going underneath, they graze the grass underneath. You get a drip line in the mornings, in the dry night times. Times of the year where there's no rain, there's a drip line at the bottom of the panels from condensation. So you get green all year round. So you get actually the sheep grazing under the panels have 30% increase in mutton yield tonnage, much higher wool quality. They jump a couple of grades in the wool quality, then lower stress, higher lambing rates. There's actually a co-benefit along with the income from the solar panels. And the same goes with vineyards. In vineyards, we have an 80% increase in berry growing, 50% increase in overall income for the farmer.
Yeah,
So
wow.
it's not a competition, it's a complementary. In fact, the
Yeah.
report from Europe talks about in areas of, I think it was Spain or somewhere where they've got, might have been southern Germany, they had a lot of agri-solar installs, like experiments, demonstration projects, and the neighbours were complaining who was going to get it next. They were competing with the other farmers to have it put on their place rather than objecting.
There you go. There you go.
Cool.
Awesome. Thank you.
Thanks, Richie.
If our farmers say agri-biodiversity hotspot, it's a little mixed organic certified place. If you're interested to come and visit, you'll find a demonstration of agri-biodiversity writ large. Whereabouts are you based? Is anyone based in Victoria?
Most of us are from Victoria and the Port Phillip Western Port catchment, a few across outer areas of Victoria as well. And Joel's obviously over in Canada.
Well, is he in Canada now or is he in Australia at the moment?
Canada.
No, yeah, I mean, yeah,
Okay.
I'll be in. I'm originally from Brisbane, and but I come back to Australia regularly, so I'll be back in March next year for a month. So
Oh,
yeah.
we'll look forward to seeing you then. Thanks.
Thank you.
Thank you.
Cool. Thank you. And I think, Joel, Agriculture Victoria, I think, have the panels up at their Tatura farm where they've been doing demonstrations up there or trials up there. So might be something
Okay.
worth looking at through AgVic. If I'm not sure if we're allowed to do a visit, but potentially we might be allowed to do a visit up there at some
Okay.
point if you
Yeah.
you know if you're interested in doing that. That's up at the Tatura farm.
Okay.
Okay, I'm going to take one more question. If anybody has any burning questions before the end, and otherwise we will finish the webinar off and put a recording out probably later this week or early next week. So just any last questions? You've got 10 seconds to put, no, less than, to put your hand up.
Yeah, there's one in the Q&A by Jeremy. I'll try and unmute Jeremy so that he can just ask the question.
If not, I'll call it out.
Jeremy, can you find the mic? I'll just read it out. Cool. All right.
I was wondering if you had any insights on using like biological products as like pest and disease management in vineyards, particularly like trichoderma and bacillus species, and like sort of how compatible they are with doing targeted foliar nutrition as well.
Yeah, I think sorry, excuse me. I think the benefit here is actually about combining the two. For that point that I was kind of making, is that the nutrition actually is essential for the plants to produce those defense chemicals, and then the biology is often what sends the triggers or the signals to induce those chemicals. So they work very much hand in glove together. The one practical concern or consideration, however, is that you do have to be a little bit careful about if you're mixing higher concentrations of nutrients, foliar nutrients, with biologicals, because if your nutrient mix is a little bit too hot or a little bit too salty, too high EC, electrical conductivity, then you can have a bit too much nutrient and it can actually have some suppressive effects over the biology. And this is a really tricky one to navigate, because don't get me wrong, the biology, your beneficial, your trichodermas, your bacillus, etc., all of those bacillus species that are helpful here, they also require those same nutrients. So biology requires essential macro micro minerals, and so there's a real enhancement that can unfold when you bring them together. But it's just about being mindful about not overdoing the nutrition, because it can become a bit too concentrated, a bit too hot, a bit too salty, and that can have some negative effects. But in order, the practical suggestion I would make here is that you can use an EC meter. And if you're using an EC meter, a guideline here would be You can use, and the EC is going to measure the electrical conductivity, so the kind of essentially the saltiness of the solution. And your guideline here is about 1.5 to 3 on an EC meter if you're measuring the millisiemens per centimeter, which would be 1,500 to 3,000 microsiemens, depending on how your setting is. But anyway, it's 1.5 to 3 or 1,500 to 3,000. That generally in that range is a very low concentration that's not going to affect the biology. So as long as the nutrients that you're mixing up, whatever they are, as long as they're not too concentrated, so in that kind of range, you'll find that they should be compatible and it should be okay. And yes, it is a good strategy because really what you're doing is providing two different mechanisms, two different modes of action. One, through supplying nutrition for the plant. Two, through providing the beneficial biology that are going to, colonize that leaf area and suppress the pathogen. But those organisms are also going to help to send various signals, communication chemicals to the plant, which helps to upregulate those defense chemicals that I mentioned in the lecture as well. So look, both Trichoderma and Bacillus subtilis is, a very famous one, particularly. They're both very famous disease fighters, and using a combination of those along with nutrition can be very effective. Yeah.
And what about, like, I'm keeping them separate at the moment, but also like timing of day, spraying, environmental conditions. Like, are you able to sort of just touch on like what conditions are like more conducive to doing like good foliar nutrition, like with water rates and that sort of thing as well?
Yep, sure. So the key thing in order to ensure that nutrition is absorbed into the leaf, you really actually want a little bit higher humidity conditions. And I know that's tricky, particularly in the vineyard world, because that creates also, high humidity, and water is also more susceptible for pathogens. But under higher humidity, the plant stomata and the micropores and the cuticle, they're more open. So the plants are able to absorb the nutrition better. So the best time to be foliar spraying is high humidity. And the magic number here is around about anything above 70% humidity, you'll find is good for foliar uptake. But you can always use the delta T. I'm sure you're probably familiar with delta T. Just type in delta T into Google Images and you'll come up with a graph which plots humidity with temperature, and it gives you an optimum green zone or an optimum area in which relative to the temperature and humidity, which you can apply foliar nutrition, and that's when it will be taken up optimally. Because if you apply in the middle of the day, and like on a hot summer's day, for example, I say a hot summer's day in Australia. You guys, I hear you're having a cold snap today, but it was only a few weeks ago you were having a heat wave too. So on a hot day where there's very low humidity, that's going to make the plant less open, and therefore it's going to absorb less nutrition. So generally the rule is going to be kind of early mornings, late afternoons, and in the evenings. And look, if you're in a vineyard context where you're worried about the wet leaves and the disease, fine, I would focus more on the early morning. I think early morning would be the priority in a vineyard context. But look, I know many other farmers in wheat and broadacre and other cash crops and the like, they will apply in the evening, like in the middle of the night, in the evenings, when it's a little bit cooler, when it's a little bit higher humidity, and you'll get better absorption of that nutrition. So yeah, definitely the time of day is really important. The key factor here is you want a little bit higher humidity. And type in delta T into Google Images, and that'll give you the perfect answer as to whether or not it's a good temperature humidity combination timing for that foliar application.
Okay, thank you.
Oh, thanks, Jeremy. Okay, we've gone sort of 20 minutes over time, so if there's no more questions, we'll call it a day. I just want to say thank you very much, Joel, for joining us from Canada, from the nice snow over there. We're getting a bit of snow apparently here in Victoria on the high country today. And also thank you to Mary for her pre-recording as well. The feedback we got for this webinar was people were interested in getting more information on how to improve plant health through integrated pest management and pest and disease management. So I'm hoping that we've answered some of those questions for people today, and go well. Thank you very much for joining us here today. There will be a recording made available in the next couple of days, which we'll send out to everybody. I will, just before we finish off, and those of you that are still online, I've just quickly shared a QR code. Like with all things, we love to have a bit of evaluation feedback. I'll also be emailing this to everybody as well. So if you don't do it now, you'll get harassed again to fill in the evaluation for us and give us some feedback on this webinar. That would be fantastic. Thank you very much, everybody, for joining us.